I hope you already had enough of potassium, sodium, and calcium. So let's switch to the most important anion, as Marriott mentioned. So my lab mainly study chloride channels. They are most abundant. Actually, you might be surprised. Actually, there are still many chloride channels. We don't know what protein or genes underlying them. And this is one of the main reasons is that actually many channels, these cation channels, share homologies. So after sequence the human genome, you know, these are the channels. But for various families of chloride channels, you don't share sequence homology. So by look at sequence, you don't necessarily know, this gene would be a chloride channel. So in the last 10 years, I've been working on trying to decoding these channels and first find out the gene, and then you could study their function. So that's kind of the main premise. So the channel I want to talk to you about today is called the proton-activated chloride channel. And we know acid is very important in physiology and disease. Many disease has acidosis in cancer or pain. Many vesicles inside of cell also are acidic. They are very important for their function. And accordingly, actually, acid can activate many kinds of receptors, GPCRs, cation channels. So one of the channels actually is widely expressed in cells, as shown here in this recording, where if you perfuse the cell with acid, you activate this large outward current. And there's also an inward current, which actually is activated. It's the A6 channel. It's also expressed. And this A6 channel can be blocked by a mineroid, which can inhibit the channel. However, the outward current is still present. So this is mediated by a chloride. And the sensitivity is not very high, so it activates around pH 5. So it was thought to be not very physiological in a way. So that's one of the reasons that people haven't been paid too much attention to the channel. And obviously that it's a mystery that what's the function, why you would have this channel in the cell surface. So in order to address that, obviously we want to identify the gene. And you could do, I wish I could Q-patch, you could recording and screening a bunch of genes, but we don't have it. And we turned into a fluorescent assay, which actually quite robust to screen chloride channel modulators for CFTR, for example. So it utilizes a very simple principle where the fluorescent protein, YP, it's a mutant version. When it's binding to anion, in particular the iodide, the fluorescence will quench. And so the idea is that it could make a cells, which the cell endogenously express the acid-activated chloride channel. And when you perfuse the acid, it will open the channel. And because the chloride channels are not very selective, and they don't have to, because chloride is only anion in the biological system, so they actually also can pass iodide. So you put iodide in the solution, then the channel is opened, iodide gets in the cell, you get a quench. And this actually is exactly what we found here. So the fluorescent traces where you add in acid, which activates channel, and iodide, which get into the cell to quench the fluorescence. And this activity is pH dependent. So this assay actually works very well. So next, we need a way to knock down gene or knock out gene to see which gene knockout would get rid of this activity. And we're actually very lucky to get this SN library where we're targeting transmembrane proteins, or 2 transmembrane proteins. So there's around 3,000 such genes in the human genome. And each gene, we have about 4 siRNA. So this is a typical traces where it's 384 well plate, and each well has 1 siRNA to a particular gene. And if you zoom in, so in most of the cases will be on the left where you get quenching because you have the channel, you add acid, the channel is open, you add iodide, fluorescent quench. But occasionally you see wells where there's less quenching, so those would be a potential hit. And so we went through the plates about 12,000 siRNAs because each gene has 4 siRNAs, 3,000 genes. And we were very excited that one of the genes actually, it's the only gene kind of pop out from the screen where 3 siRNAs are very potent to inhibiting this quenching response. And all the other hits only has one siRNA, most likely it's non-specific. It's particularly exciting because this is a gene where people haven't studied. So it's called TMEM. It's just a transmembrane protein, has two transmembrane domains. So we call the gene PAC for proton-activated chloride channel. In the meantime, Thomas Jentsch's group in Germany also did a similar screen, and they found this same gene. So it's very assuring that this likely is an essential component of this channel. Of course, before we get too excited, this is imaging assay you want to confirm in electrophysiology, and we made knockouts of 293 T cells and HeLa cells. And in both cases where you can see that the outward current is gone, but the inward current, which mediates ASICs, actually is normal. So it's not just knockout and the cells are not healthy. So we have a kind of internal control. So this is all well and good, and the question is whether the protein is essential, but is it encode the channel itself? For that, we actually, my postdoc Junhua and student James did a lot of electrophysiology, making mutations, and they found that the TM2 actually is very important for ion selectivity. So we think most likely this gene is encoding the channel itself. Obviously, we want to see whether they oligomerize and what is the kind of biochemical structure of this channel. So that kind of accumulates with a collaboration with Wei Lu's group at the Van Ender Institute with postdoc Zen, which we purified the protein and solved the cryo-EM structure of the human pack at first at neutral pH. This is actually quite challenging because each protein is about 40 kilodalton, so it's about 120 kilodalton for the whole complex. This is 2-3 years ago, and I mean, to me, to the audience, maybe just on the structure, but to us, it's very mesmerizing, obviously, because we just found the gene for in months, basically. Now we have a structure which is very gratifying. It is a channel. It's from a trimer. It has this beta sheets, globular domains, and it's kind of a beautiful structure. And we went on to solve the structure also at low pH, where we see large conformation changes, and particularly the external domain, basically there's a contraction movement, and the TM1 has a huge swing. And basically it's molecular devices where the head group has a bunch of residues. Upon protonation, it causes a collapsing movement where transducing to transmembrane domain. And combined with structure studies, we also made a lot of mutations, and we found that the pH sensing really depends by several histone residues in the external domain, and that transduced TM1 is very important. And the channel also desensitizes upon long-term treatment of low pH. So I will not get into details about how this channel works. But the main question, the main motivation when we first started this is that, what is the kind of physiological function of the channel? Why you would have a channel responding to very low pH, which typically don't happen. And this really kind of facilitated, the kind of hint is really coming from where the gene is, where the protein is localized. So when James made a stable cells, which fused with tag, and he found surprisingly that although we found some protein in the plasma membrane, but actually it has very prominent intracellular localization. And this immediately suggesting to us that the protein might have a physiological function in acidic vesicles inside of a cell. So he did a series of co-localization studies, as shown here, that he found that the PAC protein co-localized very well with the EEA1, which is the early endosome marker, and also transferrin, which is a recycling endosome. And to our surprise, actually, it's not localized to lysosome. I will get back to that. And possibly overexpressing a protein, they mislocalize. So to exclude that possibility, we actually made a antibody which can recognize the human endogenous antibody as shown on the left. The Western blot works very nice. And using this antibody, if you stain the endogenous cells, they also have vesicular localization. So it's not just because of expression artifact. He went on to find that there's an N and C terminus. So if it's going to endosome, it must, there's an endocytosis process. And we aligned the sequence. We found that indeed there is a classic YXX motif. Once you mutate those, either the tyrosine and leucine, that the localization were mainly stuck in the plasma membrane, and you get more current in the plasma membrane. So clearly that this is a channel which localized to endosomes. And this brings us to the old friend of why chloride is required in the endosome, in the vesicles. So basically in a vesicle where you have VATPase, which use ATP to drive proton into the lumen, but because the proton is charged, so you have more and more proton, it will not work. So the idea is that chloride is a counter-ion during this vesicular acidification process. Obviously, the star of this story is CLC, which Merritt alluded to. So this is an exchanger which exchanges one proton out and two chloride in. So now you have chloride to balance the charge of proton to facilitate acidification. So if you have a knockout of CLC, you get alkalization. And there's many mathematical papers predict why this is the best way to acidify vesicles. But our data suggests that indeed that actually there is a chloride channel there. So it's very surprising, but actually quite makes sense because this may be balancing effect of the CLC where pumping chloride in, and when the vesicles are too acidified, so the pack is activated, it starts to release chloride. So now we have to kind of revise back to all the mathematical papers and see, there is a channel which can kind of balance the chloride influx. So to test whether, we see the pack is localized endosome, whether the activity is there or not. So methods has been developed in the field where we can do whole cell recording, just like we do whole cell recording in the vesicles. So this is achieved by transfecting a cell with a mutant RAP5, which will enlarge the endosomes, as shown in the red. And if you co-stain with pack, you also see the pack staining. So let me show the video. So the way how it works is we will transfect the cells with yes, so you transfect the cells with RAP5 mutant, and then you have, after a day or two, you have large vesicles. Then you have a glass pipette come in. You can kind of rip up the membrane, and then release the vesicle. So the vesicle is about 3 to 5 micron, and then those vesicles, then can be recorded. And this method is developed by Haoxin from University of Michigan and also Dejian at UPenn. So they are very helpful to help us to establish this recording protocol. It sounds very, it's very difficult, and students very, persistent. I cannot do that, so it's very challenging. Nevertheless, the first experiment we do is that, if you patch through the vesicle, right, do you see acid activated current? And indeed, that was we'll see. So now, in the pipette solution, you would have low pH because that's kind of opposite of the extracellular, right? So we also see a very similar current with auto-rectifies. That's very nice. And this current, if you do change in solutions with low chloride inside, then you don't see the current. So basically, it's a chloride efflux from the lumen. And this current also depends on low pH because if you use neutral pH, then you don't see the channel activity. So to test whether it's mediated by PAC, we of course we record the knockout cells, and indeed that in knockout cells we don't see this activity anymore. So this suggests that the PAC mediates acid-sensitive endosome chloride currents, and the prediction is that if you knock out PAC, you might have more chloride in the endosome, for example. So this, doing this, we collaborate with Christian Lab at the University of Chicago. So she developed this DNA-based ratiometric chloride sensor. So the idea is that it's DNA-based, there's a dye hooked with those DNA. So the red dye is not sensitive to chloride, nor pH. And then you have green dye, which it will quench if you have higher chloride. So the red versus green, and then they have another component where it will bind to transferrin, essentially. So if you feed the cells with this sensor, the uptake into the into the endosome, and then you can measure the ratio. So the higher the ratio is, the higher concentration of chloride is. And indeed, that she found that the PAC knockout cells have increased endosome chloride concentration. And another prediction is that if you have increased chloride concentration, it will facilitate the proton pumping, and you would have a lower pH. And that's exactly James found, where he used transferrin conjugate with dye. One is pH sensitive, one is non-sensitive, and you can get a ratiometric measurements. And in both 293 cells and HeLa cells, he found that knockout cells are more acidified. And he did the opposite experiment, where if you have a stable cell, you overexpress PAC, and then you find a alkalization phenotype, where this alkalization actually dependent on PAC localization. So if you mutate in those two residues, then it's no longer work. So this is kind of a summary that even though we get into this, it's a whole cell recording, there's an acid active current, but actually we get a big surprise, because once we figure out the gene and we found that actually they translocated, traffic into the endosomes, where it really adding another component to the system, where people thought that you only have, CLC to pump chloride. So this actually, there is another mechanism involved to regulating acidification. And Yench group also show that this is also important for regulating volume of vesicles, because when chloride leaves, not only the charge change, but also its osmolates, so it shrinks the vesicle. And two of my students, Henry and Nick, they have posters about what kind of implication of the physiology in regulating regulating cell. So if you still have time, you can check on them. So in the last couple of minutes, I just quickly ran through another channel we study called the volume regulated anion channel. I think Stefan talked about this this morning. And again, this is actually the first channel we screened. So there's many candidates before, so we did an unbiased genome-wide screen. And instead of treating cells with acid, you just treat cells with hypertonic, so you get a quenching. As shown here, that in scrambled siRNA, you had high quenching because the channel is intact. And the DCPIB is an inhibitor for this channel, so you get the window. And this is a secondary screen after primary screen. And there's one hit stands out, is this leucine rich repeat containing 8-family A, so we also call SWELL. And coincidentally, that Yench group also did the same screen, and they also found the same gene. So we're all kind of in sync. So as Stefan mentioned, that cells don't need, there's no swelling going on most of the time, but actually the channel can be activated modestly by many diverse stimuli. For example, there's GPC activation, apoptotic drugs, and all the other metabolic stress, for example. And what this channel stands out is really that it's a large pore channel. So it's not only chloride, but also many other kind of stuff. For example, cancer drugs can get into cells through the channel. We don't know how, but we think there's a little bit activity. So it's like more like a transporter. So for us, we were involved to show that, for example, in astrocytes, they can release glutamate from this cell, through the channel to regulate cell transmission. And we were also involved in a study where it showed that the CGAP, which Stefan mentioned, that they can uptake through the channel. So one story we're working on is actually ATP, because inside of cells, that ATP concentration is pretty high, about 5 to 10 millimole. As Diane was talking yesterday, that the nerve endings can release ATP. So this channel actually can release ATP. So as shown here, that if you treat cells with hypertonic, you had huge ATP efflux. And this is dependent on SWELL1. So if you knock out, it's gone. And also if you treat an inhibitor, there's also a decrease. So clearly, it's hypertonic can activate the channel and release ATP. You can even see that by electrophysiology. So if you exchange the chloride in the pipette with only ATP, which anion, so you still see an inward current where it's basically mediated by the efflux of ATP. And this can be rescued by if you express the cDNA. And obviously, what is the biological context of here? So we turned into the microglia because purinological singlet is important for microglia activation and inflammation. And indeed, that if you have microglia cells, if you treat it with hypotonic, you get this classic SWELL current, which can be inhibited by the blocker. And more importantly, the channel can also be activated by other kind of inflammatory mediators, for example, S1P, where 10 nanomole can also activate a very robust current. And it can also induce ATP release. My student Jia Chen had a poster on this, basically that we choose a model system where it's neuropathic pain, where in the resting state after nerve injury, where the channel is activated by various inflammatory response, for example, S1P, for example, and those cells will proliferate and they activated, and then they will have inflammation, for example, and activates neuronal excitability, which will cause pain. And she used both genetic, which we have a knockout, conditional knockout, and also pharmacological. She used the same screen, the quenching assay, get a drug, dicumera, which can, it's a new inhibitor for the channel. And in both cases that she found that either genetic or pharmacological inhibition of SWELL can reduce ATP and reduce inflammation and also dampens the neuronal activity while causing preventing the neuropathic pain. So I encourage you to talk to her about the study. So I think I acknowledge all the people who did the work. And of course, we have collaborators with the structure with the Wei Lu's group at the Vander Institute and Yun Guan's group at the Johns Hopkins with a lot of pain study. And also the funding sources. And I would like to take any questions. Thank you.
Thank you. While we're waiting, are there any human mutations of PAC that are known right now? And what's the phenotype in humans?
Yeah, I'm still waiting for the phone call.
Oh.
For the SWELL, there's some links for immune system. Okay. But I think that if there is, must be very rare, rare situation. Maybe with exome sequencing, maybe there will be more being reported. Thanks. Yes, Colin.
Just a quick question. At the beginning.
Yes.
At the beginning with your screen.
Yeah.
You're measuring chloride entry.
I'm measuring chloride influx
into the cell.
Yes.
So if PAC is on internal membranes,
yeah.
How is it affecting the transmembrane flux? Or is some of the PAC getting to the surface membrane as well?
Yes, yes. So again, this is an interesting point where the channel was being described as a plus membrane channel, right? But if you do whole cell recording, you see this current. It's a trafficking thing. We don't know why it's substantial amount of it in the plus membrane. So that's, it's a little bit but my understanding that a lot of the endosomal or vesicle channels will be, transiently in the plus membrane. So that's a possibility. Yes, Mark.
I must have missed it if you said it, but it looks like a full channel is a trimer. Is that right?
Yes, yes.
Okay. And then like piezo, is there any correlation with the way piezo works to sense stretch?
Piezo?
Yes. So how does this channel sense swelling?
Yeah. So the trimer I'm referred to is for the PAC it's sensing for acid. Yeah.
Oh, I'm sorry.
So there's a constriction of the external domain. Actually, very interesting. Actually, A6 channel is also a trimer and looks very similar, but there's no sequence homology. So there might be something going on. We don't quite understand. There's some way why this type of structure is good for the pitch sensing.
Any other questions? Have you made a knockout of the PAC or?
Yeah, we made a knockout, and it's alive. Yeah, there's some neurological phenotype. So we were characterizing.
Thank you.
Sure.